mice who are not been irradiated, over time, over several trials, take less and less time to find that pedestal because they remember where that pedestal was when they're placed in the water bath. In contrast, mice that who undergo cranial radiation do not have that memory and do not improve the time it takes for them to find that pedestal. And so they continue to circle around the water bath before they can find that pedestal. To round out the story, at least from a preclinical perspective, this is a very interesting study done at UC Davis, and I think was mentioned by Dr. Gattumaneni. But in this experiment, they actually treated the rodent model with cranial radiation, and then 2 days later actually implanted neural stem cells that they had grown in vitro in culture into the hippocampus. And not only did they notice that that engraftment led to the development of hippocampal neurogenesis, but also completely reversed the memory-related decline, the memory toxicity of cranial irradiation. This is interesting, but obviously not ready for clinical use, but it does raise the idea that perhaps, again, the hippocampal neurostem cell compartment might be very sensitive to radiation. So we proposed the opportunity to use advanced radiotherapy techniques, and specifically intensity-modulated radiotherapy, to spare the hippocampal dentate gyrus of significant doses of radiotherapy during whole brain or cranial irradiation, with the idea that we may be perhaps lowering the dose to the neurostem cell compartment that is memory-specific and radiosensitive, preserving its normal neurogenic fate, and perhaps preserving memory function after cranial irradiation. I mentioned this slide earlier. We sought to provide some preliminary observations through this prospective observational analysis and observed that dose to 40% of the hippocampi was significantly associated with decline in a memory test that is very similar to the Hopkins verbal learning test that I described earlier. So we had this interesting analysis, and at the same time we were creating a number of dosimetric exercises to use IMRT to see if we could spare the hippocampi during whole brain radiotherapy. And now that we had this threshold of dose to 40% less than 10 gray, we went back to our IMRT dosimetric exercises and observed that modern IMRT techniques, at least the ones that we published, seem to lower the dose to 40% of the hippocampus to 10 gray or less, suggesting that modern IMRT may sufficiently spare the hippocampi to provide a clinical benefit. But when sparing the hippocampi, so when we first proposed hippocampal avoidance, we decided to do so within the context of whole brain radiotherapy for brain metastases, in part because at that time the MD Anderson study demonstrated decline in memory function, HVLT, had just been presented at ASTRO as a plenary session. And we thought, well, what a better situation or clinical context in which to test this hypothesis. But obviously, when sparing the hippocampus of significant therapeutic doses of radiation, there is a risk of the emergence of a new metastasis within the hippocampus because we're not delivering therapeutic doses of radiotherapy. So we sought to estimate this risk, and we did a multi-institution analysis of 371 patients who presented with brain metastases and observed that 8.6% of those patients presented with a brain metastasis within 5 millimeters of the hippocampus, what we call the hippocampal avoidance region, but none of them presented with a metastasis within the gray matter of the hippocampal dentate gyrus.